What if I told you there's an organism growing right now on this planet that can eat plastic? Not break it down slowly over centuries, like nature struggles to do. Eat it, digest it, consume it as food, the way you eat a meal. What if I told you the same type of organism was found growing inside the most radioactive place on Earth, the Chernobyl nuclear reactor, where radiation levels are so extreme that robots sent in to clean up the disaster broke down within minutes, where no living thing should be able to survive. And yet this organism was not just surviving, it was thriving, feeding on the radiation itself, using nuclear energy the way plants use sunlight. What if I told you another species of this same organism can break down petroleum, crude oil, the toxic hydrocarbons that destroy ecosystems when tanker ships spill millions of gallons into the ocean? This organism eats it, cleans it, turns contaminated soil into fertile ground in weeks, not years, not decades, weeks. And what if I told you that despite all of this being documented in peer-reviewed science from Yale University, Stanford, the University of Nottingham, and dozens of other institutions, despite TED Talks watched by millions, despite thousands of published studies, the total global funding for this technology is almost nothing compared to the industries that profit from the problems it solves. The plastic industry is worth $600 billion a year. The nuclear waste cleanup industry charges governments billions. The oil spill remediation industry is a multi-billion-dollar business. These industries do not sell solutions. They sell management. They sell containment. They sell the slow, expensive process of dealing with disasters that never fully get cleaned up. And then a mushroom shows up, grows in 2 weeks, costs almost nothing, and eats the problem. This is not science fiction. This is not a hypothesis. This is not a theory waiting to be proven. This is mycology, the science of fungi. And what I'm about to show you in this video will fundamentally change the way you see the world beneath your feet. Stay with me. Every word of this is documented. Every claim has a source. And by the end of this video, you will understand why the most powerful cleanup technology on Earth is being grown in laboratories and ignored by the industries that should be using it. In 2011, a group of students from Yale University traveled to the Amazon rainforest in Ecuador on a research expedition. They were collecting samples of endophytic fungi. These are fungi that live inside the tissues of plants without causing harm. The students were cataloging species as part of a biodiversity study. Routine academic work, nothing groundbreaking. Or so they thought. Among the dozens of fungal species they collected, one stood out, a species called Pestaloteuopsis microspora. When they brought it back to the laboratory at Yale and began testing its properties, they discovered something that had never been documented before in the history of science. This fungus could digest polyurethane. Polyurethane is one of the most common and most persistent plastics on Earth. It is used in everything from shoe soles to furniture cushions to building insulation to automotive parts. It is in your couch right now. It is in the mattress you sleep on. It is in the insulation inside your walls, and it does not biodegrade. Not in any meaningful time frame. A polyurethane product thrown into a landfill today will still be there in 500 years. In 1,000 years. Potentially longer. We do not actually know how long polyurethane lasts in the environment because none of it has ever fully broken down since we started manufacturing it. Until Pestaloteuopsis microspora. The Yale team published their findings in the journal Applied and Environmental Microbiology in September 2011. The paper documented that this fungus could break down polyurethane as its sole carbon source. Let me explain what that means in simple terms. Most organisms that decompose materials need multiple nutrients to survive. They need carbon, nitrogen, various minerals, and water. They break things down as part of a larger metabolic process that requires many inputs. Pestaloteuopsis microspora can survive on polyurethane alone. It does not need soil. It does not need other nutrients. You can put this fungus in a container with nothing but polyurethane plastic, and it will grow, it will feed, it will reproduce, and it will convert that plastic into organic matter. Even more remarkably, the Yale researchers found that this fungus can do this in anaerobic conditions. That means without oxygen. This is critical because the bottom of landfills is an anaerobic environment. There is no air down there. Most decomposition processes require oxygen. This fungus does not. So picture this. The billions of tons of polyurethane plastic buried in landfills around the world, sitting in airless darkness where nothing can break them down. This fungus can be introduced into that environment and it will eat the plastic. In the dark, without oxygen, converting it into harmless organic compounds. The Yale discovery made headlines briefly. Science magazines covered it. A few news outlets picked it up. And then it faded from public attention. No major corporation invested in scaling the technology. No government launched a national program to deploy plastic-eating fungi in landfills. The paper has been cited over 300 times by other researchers, but real-world application remains minimal. Meanwhile, every single year, the world produces over 400 million metric tons of new plastic. 400 million metric tons. And less than 9% of all plastic ever manufactured has been recycled. 9%. The rest is in landfills and oceans and rivers and soil and in the bodies of animals and humans. We are drowning in plastic, and a fungus that eats it was discovered 13 years ago in a university laboratory by students. But that is only the beginning of this story, because what was found growing inside Chernobyl makes the plastic-eating fungus look ordinary. On April 26, 1986, reactor number 4 at the Chernobyl nuclear power plant in Ukraine exploded. It was the worst nuclear disaster in human history. The explosion released 400 times more radiation than the atomic bomb dropped on Hiroshima. An exclusion zone covering over 2,600 square kilometers was established. Over 300,000 people were permanently evacuated from their homes. The area was declared uninhabitable for the next 20,000 years. 20,000 years. The concrete sarcophagus built to contain the destroyed reactor was designed to keep the radiation sealed inside. But within years, cracks began forming. Water seeped in. Radiation levels inside the structure remained so extreme that human beings could not enter without risking fatal exposure within minutes. In 1991, 5 years after the disaster, a team of researchers sent a remote-controlled robot into the ruins of reactor number 4. The robot carried cameras and sample collection equipment. Its mission was to survey the interior of the most radioactive building on Earth. What the cameras showed was impossible. Growing on the walls of the reactor, on the concrete surfaces bombarded by levels of gamma radiation that would kill any known living organism, a black fungus was spreading, not dying, not clinging to life in some diminished state, spreading, growing, thriving. The researchers were stunned. They collected samples using the robot and brought them back to the laboratory for analysis. What they discovered over the following years would challenge everything biology textbooks taught about life on Earth. The fungus was rich in melanin, the same pigment that colors human skin, but in these fungi, the melanin was doing something that had never been observed before in any living organism. It was converting gamma radiation into chemical energy. The process was named radiosynthesis, and it is exactly what it sounds like. Just as plants perform photosynthesis by converting sunlight into energy through chlorophyll, these fungi were performing radiosynthesis by converting nuclear radiation into energy through melanin. They were eating radiation. In 2007, researchers at the Albert Einstein College of Medicine in New York published a landmark study in the journal PLOS One that confirmed this mechanism. The lead researcher was Dr. Arturo Casadevall, one of the most respected microbiologists in the world. Casadevall and his team tested 3 species of melanin-rich fungi: Cladosporium sphaerospermum, Wangiella dermatitidis, and Cryptococcus neoformans. They exposed these fungi to radiation levels 165 times higher than normal background radiation, levels that would damage or destroy most living cells. Yet the fungi did not just survive, they grew faster. The irradiated fungi showed significantly increased growth rates compared to control groups that were not exposed to radiation. The melanin in their cell walls was absorbing the radiation and converting it into metabolic energy that fueled their growth. Let me say that again because it is genuinely one of the most extraordinary discoveries in modern biology. These fungi grow faster when you blast them with radiation. They are not just radiation resistant, they are radiation powered. Casadevall's team proposed that radiosynthesis may have been one of the earliest forms of energy harvesting on Earth. All billions of years ago, when the planet was young and surface radiation levels were much higher than they are today, melanin-rich fungi may have been among the first organisms to colonize the Earth. Before photosynthesis even existed, life may have started not with sunlight, but with radiation. Now think about the implications of this discovery for nuclear waste management. Right now, there are over 440 nuclear power plants operating in 32 countries around the world. These plants have been generating radioactive waste for over 70 years. High-level nuclear waste remains dangerously radioactive for tens of thousands of years. There is currently no permanent solution for storing this waste. Governments spend billions of dollars per year on temporary containment facilities, billions more on cleanup operations at decommissioned sites. The total global cost of nuclear waste management is estimated at over $100 billion and growing every year. And there is a fungus that eats radiation. In 2018, researchers proposed sending melanin-rich fungi to the International Space Station to test whether they could serve as a biological radiation shield for astronauts during deep space missions. The concept was simple. Grow a layer of this fungus on the walls of a spacecraft and let it absorb the cosmic radiation that threatens crew members during long missions to Mars. In 2020, that experiment actually happened. Cladosporium sphaerospermum was grown aboard the International Space Station. The results showed that a layer of this fungus, just 1.7 millimeters thick, reduced radiation levels by nearly 2%. A thicker layer of approximately 21 centimeters could theoretically reduce radiation to levels safe for human habitation. NASA published the results. Scientists around the world took notice. And then nothing happened. No major funding program was launched. No government announced a plan to deploy radiation-eating fungi at nuclear waste sites. No corporation invested in scaling this technology for spacecraft shielding. The nuclear waste management industry continued charging governments billions for concrete and steel containment structures. The space radiation shielding industry continued developing expensive synthetic materials. And the fungus that eats radiation continued growing in the ruins of Chernobyl, unbothered, unfunded, undeployed. But we are not done yet, because there is a third ability, and this one was demonstrated not in a laboratory, but on one of the largest stages in the world, by a man the scientific community considers one of the greatest mycologists who ever lived. In 2008, a mycologist named Paul Stamets walked onto the TED stage and delivered a presentation that would be watched by over 6 million people. The title of his talk was 6 Ways Mushrooms Can Save the World. And he was not exaggerating. Stamets had spent over 40 years studying fungi. He had published multiple books. He held patents on mycological technologies. He had worked with the United States Department of Defense on classified biodefense projects. He was not a fringe scientist. He was not a conspiracy theorist. He was one of the most credentialed and respected mycologists on the planet. And he stood on that stage and told the world that fungi could clean up the worst environmental disasters humanity has ever created. One of the most powerful demonstrations Stamets presented involved petroleum contamination. He described an experiment conducted in partnership with Battelle Laboratories, one of the largest private research organizations in the United States. Stamets took 4 piles of soil. Each pile was heavily contaminated with diesel fuel and petroleum hydrocarbons, the kind of contamination you find at oil spill sites, industrial waste dumps, and abandoned gas stations. Toxic soil that kills plants, poisons groundwater, and remains contaminated for decades. One pile was left untreated as a control. One pile was treated with chemical remediation agents, the standard industry approach. One pile was treated with bacteria-based bioremediation, another standard approach. And one pile was inoculated with oyster mushroom mycelium. Stamets simply mixed mushroom spawn into the contaminated soil and covered it with a tarp. 6 weeks later, they checked the piles. The control pile was still black and toxic, dead, nothing growing. The chemically treated pile showed some improvement, but remained largely contaminated. The bacteria-treated pile showed moderate improvement. The mushroom pile had exploded with life. The mycelium had consumed the petroleum hydrocarbons, broken them down into simpler organic compounds. The toxic black soil had been transformed into rich brown earth. And covering that earth were hundreds of oyster mushrooms, some of them over 12 inches in diameter. But here's what happened next that stunned everyone. Insects found the mushrooms. Insects attracted birds. Birds deposited seeds in their droppings. Within weeks, the mushroom pile was covered in plants and grasses. A miniature ecosystem had spontaneously emerged from what had been toxic dead soil just 6 weeks earlier. The petroleum was gone. The toxicity was gone. Life had returned in 6 weeks, using mushrooms. Stamets called this process mycoremediation, and the science behind it is straightforward. Fungi are the master decomposers of the natural world. They have been breaking down complex organic molecules for over 1 billion years, long before humans existed, long before petroleum was ever pumped from the ground. Fungi were already equipped with the enzymatic machinery to disassemble hydrocarbon molecules, because hydrocarbons are not alien to nature. They are carbon chains. And carbon chains are what fungi have been eating since before dinosaurs walked the earth. Petroleum is essentially ancient organic matter compressed underground for millions of years. To a fungus, it is just a very old meal. The enzyme systems that fungi use to break down wood, dead plants, and other organic matter are the same enzyme systems that break down petroleum. Lignin peroxidase, manganese peroxidase, lacase. These enzymes are molecular scissors that cut complex carbon structures into simple, harmless compounds. Stamets demonstrated this repeatedly, in laboratories, in field tests, in partnership with government agencies and major research institutions. The results were consistent and reproducible, and the cost was almost nothing. So let us add this up. A fungus discovered by Yale students that eats plastic, a fungus found in Chernobyl that eats radiation, a fungus demonstrated by one of the world's leading mycologists that eats petroleum and restores dead soil to life in 6 weeks. 3 of the most devastating environmental problems facing humanity: plastic pollution, nuclear waste, oil contamination, each one a multi-billion-dollar crisis, each one considered virtually unsolvable by conventional methods. And fungi can address all three, naturally, cheaply, in weeks. So why is nobody using this at scale? Let us follow the money. The global plastic waste management industry is worth over $50 billion per year. This industry profits from collecting, sorting, transporting, and burying plastic in landfills, or burning it in incinerators. They do not profit from eliminating plastic. They profit from managing it endlessly, expensively, inefficiently. A fungus that eats plastic in a landfill without human intervention is not a business opportunity for the waste management industry. It is an existential threat. The global nuclear decommissioning and waste management industry is worth over $100 billion. Companies charge governments enormous fees to build concrete containment structures, monitor radiation levels, and manage waste storage facilities that must operate for thousands of years. These are contracts that last generations, revenue streams that never end. A fungus that absorbs radiation and converts it to biomass is not a business opportunity for the nuclear waste industry. It is an extinction event. The global oil spill cleanup industry generates billions in revenue every time a tanker runs aground or a pipeline bursts. Companies like those contracted after the Deepwater Horizon disaster in 2010 charged BP over $65 billion in cleanup costs and damages, $65 billion for one spill. A bucket of mushroom spawn that costs a few dollars and cleans contaminated soil in 6 weeks is not a business opportunity for the oil cleanup industry. It is a nightmare. Now I want to be very clear about something. There are legitimate scientific challenges to deploying mycoremediation at scale. Fungi require specific temperature and moisture conditions. Different contaminants require different fungal species. Scaling laboratory results to real-world environments is complex. These are real obstacles, but they are engineering problems, not fundamental science problems. The science is proven, the biology works. What is missing is funding, investment, political will, and the courage to challenge industries that profit from problems remaining unsolved. Paul Stamets has said publicly and repeatedly that the funding available for mycoremediation research is a tiny fraction of what is needed. Universities studying plastic-eating fungi operate on shoestring budgets. The Chernobyl radiotrophic fungi research receives minimal government support. The mycoremediation companies that do exist are small startups competing against billion-dollar waste management corporations with armies of lobbyists. The technology is not the problem. The economics are the problem. And the economics are controlled by people who make money from the status quo. Let me leave you with one final thought that puts all of this into perspective. Beneath your feet, right now, there's a network of fungal mycelium running through the soil. Scientists call it the mycorrhizal network. Some call it the wood wide web. This network connects trees and plants across entire forests, allowing them to share nutrients, send chemical warning signals about pest attacks, and support weaker members of the ecosystem. This network has existed for over 1 billion years. It was here before humans, before mammals, before dinosaurs, before the first plant ever grew on dry land. Fungi were already here, breaking things down, cleaning things up, recycling the dead into food for the living. Fungi are the original recyclers of planet Earth. Oh, they have been solving decomposition problems for longer than any other organism in existence. They have had 1 billion years of evolution to perfect their enzyme systems. 1 billion years of practice breaking down every complex molecule nature has ever produced. And now in the blink of an evolutionary eye, humanity has created 3 new complex molecules that are destroying the planet. Plastic, nuclear waste, petroleum contamination. And the fungi looked at these problems the same way they have looked at every dead tree and fallen leaf for 1 billion years. As food. We did not invent the solution to plastic pollution. We did not engineer the answer to nuclear waste. We did not design the technology to clean oil spills. The solution was already here, growing in the soil, growing in the Amazon, growing on the walls of Chernobyl. It was always here. We just refused to use it. 400 million metric tons of new plastic produced every year. $100 billion spent on nuclear waste containment, $65 billion charged for a single oil spill cleanup, and a mushroom that grows in 2 weeks could help solve all 3. But mushrooms cannot lobby Congress. Mushrooms cannot buy advertising. Mushrooms cannot hire public relations firms. Mushrooms cannot donate to political campaigns. And mushrooms cannot generate recurring revenue for shareholders. And in a world where solutions are only deployed when someone profits from them, a free solution growing in the dirt will always be the last option considered. Unless enough people learn about it, unless enough people demand it, unless enough people share this information and force the conversation into the mainstream where it belongs. The fungi have been waiting for 1 billion years. They are patient. They are everywhere. They are ready. The question is whether we are smart enough to use them, or whether we will keep paying billions to manage problems that mushrooms could eat for breakfast. Some knowledge grows in the dark, just like fungi. It is time to bring it into the light. If this video changed the way you see the world beneath your feet, share it with someone who needs to hear this. Hit subscribe and turn on notifications, because the next video uncovers an ancient material that scientists at MIT just proved is stronger than anything we can build today. 2,000 years old, and we still cannot replicate it. Drop the word mycelium in the comments if you made it to the end. I read every single one. I will see you in the next one.